If there is one thing we learned from the coronavirus pandemic, it is this: we are all connected. The world was at risk and millions were dying. But at last, collectively, we found the tools to save ourselves. And we have to do it again. This is not the time to be complacent, as an even greater health risk is on the rise. Antibiotics are failing. If we don't find alternatives now, we will return to a world before antibiotics, when millions of humans died of simple infections. Ben Chan, world-renowned scientist at Yale University, is convinced that a tiny virus that you can find in bacteria-contaminated waters will save humankind.
Hey, Martin.
Hi.
Hey. How's it? Good to see you guys again.
You can
Do you want me to? I'm okay with that, but
Let's just wait.
you want me to?
Yeah.
It's the regulations. Okay.
How do you usually collect your samples?
Hand sanitizer and a little scooper.
Trillions of microorganisms live in and around us. Among them, the most significant source of genetic diversity are bacteriophages, or phages for short. These tiny viruses infect and kill bacteria. Harmless to humans, they operate by injecting their genetic material into bacterial cells, where they replicate exponentially and cause the bacteria to burst open. Although they are the most abundant entity on Earth, we know very little about these nanoscopic killing machines. Every day, they kill 40% of bacteria on our planet. They keep bacteria in balance. Without them, we would not be able to exist.
You should have brought the gumboots, right? Yeah.
I'm right behind you.
I think for me, my shoes are very appropriate.
I think people don't appreciate that this can be a extreme sport over here, huh?
Mm-hmm.
You have to be interested in sewage and sort of gross stuff if you're interested in bacteria that cause human diseases. Because wherever those bacteria are, that's where we're going to find the phages that can kill them. For any bacteria that's out there, there is a bacteriophage. It's just, if we want to try and use it as a therapy, our goal is to try and find the right one for the right bacteria.
From this side?
Even just there, it's probably okay, because it's all sort of scooping together, and there's like a, I don't know, a dead something in the water there. It's one of my favorite parts of the job, because we're always finding new phages. Never found the same phage twice. With every new phage is a new potential treatment. Yeah, lots of protein in the water. Yeah, I mean, that'd be great to sample from.
A woman there, she was asking me what they have done wrong. You know, they think it's the government.
Yeah, exactly, right?
Yeah, that's what she was asking
That's
me
why I
about
usually don't wear like this kind of stuff, because people are like,
Yeah.
why are you here? Like, what are you studying? Is that?
Yeah.
Yeah.
So I think they are with the police.
Yeah. Klebsiella also.
Klebsiella. Yeah.
And what's 51137?
That's
ST.
ST, yes. That's Cinetobacter strain.
Nice. Phage therapy has potential to produce some pretty dramatic changes in the way we treat infections and the way we produce pharmaceuticals. A handful of companies develop all of the antibiotics that are available. Where phages because of the lack of intellectual property, they're open for anybody to use. Phages could be produced for a fraction of a penny for a dose. Dedicated labs all around the world could create phage products, and they're no longer dependent on these large companies.
Drum rolls. Yeah. So we'll start by organizing them.
Yeah.
We had 2 E. colis, 2 Klebsiellas.
These are me.
Yeah, these are.
I think you might have won. I got like 12.
I got a lot.
More than 12?
More than 12. 2 minutes to count.
You won.
Yeah.
Congratulations. Oh, that one's awesome.
Yeah.
You got lots of good ones.
Fascinatobacter.
Yeah.
Yeah.
With a nice halo.
It would be interesting to check out cholera.
There's a cholera outbreak in Kibera.
Yeah.
If you have strains, we should definitely screen the samples on it. And if not, then we'll make it work somehow.
How can you not get infected by cholera in places like Kibera? Access to vaccination is limited. Clean water and sanitation are a rare commodity you must pay for. Healthcare facilities are overcrowded. Medications are often not available and very expensive. Most patients can't afford to use antibiotics as prescribed. What is available is often an expired product, disposed of by Western countries and exported here. The misuse and overuse of antibiotics have led bacteria to mutate into superbugs. In a post-antibiotic world, all of our scientific advances, organ transplantation, and chemotherapy will be erased. The World Health Organization estimates that antimicrobial resistance was directly responsible for 1.27 million global deaths in 2019 and contributed to almost 5 million deaths. We knew about these risks right from the start of penicillin, but with antibiotics being the wonder drug that saved millions of lives, we ignored the danger of multi-drug resistance. Phage research is thriving worldwide and has led to many international collaborations. Toby Nagel, founder of the NGO Phages for Global Health, has come to Nairobi to help advance phage therapy.
I had spent 15 years working in the drug development field, making drugs that typically cost $1 billion and maybe take 10 years to make. But on the side, I was always consulting in developing countries. These countries typically get the drugs, say for COVID or for other things, after industrialized nations do. We want to reverse that. We want to have local drug development, even with all the measures we're putting in place. We are going back to a pre-antibiotic world. It's a matter of how quickly we can stem the tide.
Lillian, this campus is beautiful.
It is very beautiful.
It really is.
You know, the campus was designed by the Japanese. They sponsored this campus.
Yeah.
So this is the Center for Microbiology Research.
Great.
So this is where we live. They're our hosts.
Great. Thank you. Thank you, Eric.
Are your people here?
Yeah, they're here.
So welcome to our microbiology lab. Our bacteriophage team are here. So guys, I told you our visitors are coming. Here they are live. This is Dr. Toby Nagel, who's the CEO of Phages for Global Health. And then Dr. Min Min Yen is the CEO as well of Phage Pro. And then Dr. Ben, as you've heard, is a clinician from Yale University in the US. He just told us he's treated between 70 and 100 patients using phages. So he's really here to show us that it's possible. It's not witchcraft.
It's to see if it's able to lyse.
Antimicrobial resistance is probably the biggest global health threat that mankind will face. For COVID, I think in 2019, 3 million people died, and it brought the world to a standstill. Now, what will we do with 10 million people dying a year? The bulk of those deaths will happen in sub-Saharan Africa. We don't always have access to the latest medicines. We don't have access to the latest vaccines. So even a mildly multi-drug resistant infection could mean death to many patients. So we started a project looking at bacteriophages, viruses that could kill these multi-drug resistant bacteria just from our environment. So we really have an opportunity to pit biology against biology, genetics against genetics.
Do you find it difficult to get any of the reagents that you need, or?
Supplies is a major issue here.
Right.
Even though we can import things, our costs are 3 times maybe what it would be in Europe or the US when you're close to the vendors and you don't have these markups.
Simple syringe filters, they cost tenfold more here than in the US.
Yes.
Yes, they do.
Easily.
Yes, they do.
The hundreds of phages isolated at Kemry are shared with the Walter Reed Army Institute of Research in Bethesda, Maryland. Michael John Nikolic is the lead researcher.
So this is our freezer room. You know, we keep all of our bacterial stocks in here. So we're now storing our phages at -80 as well. We have our frozen phage stocks. Some of these are frozen samples that we receive from our overseas labs. So we keep a copy at 4 degrees. We keep a frozen copy as well.
I worked in vaccine research for about a decade, working in biodefense after the fall of the Soviet Union, trying to focus on counterproliferation of agents of mass destruction, biowarfare, bioterrorism agents. Going all the way back to the beginning of our engagement in Iraq, we had infections with multi-drug resistant pathogens, and these led to loss of limb or loss of life. The mission of my team is to develop off-the-shelf antimicrobial products. In order to do that, we really need to have a diversity of phages. We started in 2020, establishing the global networks, working with our lab in Kenya, our lab in Bangkok, and our lab in Tbilisi. Already we're finding phages that we were not able to collect in all the years here.
One of the Walter Reed Institute's main partnerships in phage therapy is with the Eliava Institute in Tbilisi, in the former Soviet Republic of Georgia. Over the years, the Institute has played a significant role in advancing research on bacteriophages. Eliava has the most extensive bacteriophage collection in the world. The collection hosts over 1,000 phages active against human, plant, and animal bacterial pathogens. Despite the rise of antibiotics, the Institute continued its work on phage therapy. It is one of the few places where you can buy phages over the counter. Patients from all over the world with serious bacterial infections, especially those that are resistant to antibiotics, make the long trip to Eliava to receive phage therapy. Bacteria have been growing and multiplying for almost 4 billion years, since life first emerged on Earth. There are more bacteria in our bodies than human cells. Many of them are beneficial, keeping our microbiome in balance. Antibiotics disrupt the balance of the microbiome by killing almost all bacteria they encounter, both beneficial and harmful. To survive these attacks, bacteria evolve, become more resistant to antibiotics, and become hypervirulent superbugs. Even our last resort antibiotics have started to fail. Gaithersburg, Maryland has emerged as a micro-hub for phage research. One of the leading biopharma companies is Adaptive Phage Therapeutics. The Walter Reed Army Institute provides many of its phages. CEO Greg Merrill shows us around.
Here we are in the executive offices of Adaptive Phage Therapeutics, and one of the things that we've done is a cartoon illustration of the life cycle of phage. You can see a phage binds to the surface of the bacteria, and here we see the DNA in the phage being injected into the bacteria, and here the phage are being assembled inside the bacteria, and finally the phage is able to lyse the bacteria and release the phage into the environment, and now the phage is floating around the environment just like at the beginning of this lytic cycle. Phage presents a wonderful opportunity to leverage nature's bacterial killer. Phage are the most prolific killers of bacteria on Earth. So what you're seeing here is a petri dish, and where we're able to find phage from the environment that's able to kill the bacteria, those phage will create these clear circles. It's really a zone of death of the bacteria. We're then able to harvest the phage that's able to kill the bacteria.
APT applies cutting-edge automation to the virtually infinite supply of phages in nature. Its business model is a bit like Coca-Cola's. It patents its phage bank and then leases vending machines stocked with those APT phages to hospitals around the country.
One of the big challenges in getting the financial support for phage therapy is that historically, antibiotics have been a failed business model. That failure stems from the very basic biological fact that bacteria evolve resistance. That's driven a lot of new antibiotic companies into bankruptcy. What we're doing with phage completely turns that on its head. We can train the phage by exposing it to the bacteria over and over again until we start to see phage evolve that can kill that bacteria. That's actually a relatively fast process. That new phage is manufactured, put into the collection, and now we've covered that bacteria. So we're always able to expand the coverage of our phage collection.
High tech has its price. APT estimates that a treatment with their phages will cost around $20,000. A profitable business model. Recently, APT has merged with Israeli biotech firm BiomeX for $50 million.
We are currently receiving between 1 to 2 requests every day for emergency or compassionate use of the phage. Phage therapy is not currently approved by our FDA. It's going to still take a few years before we're able to get the regulatory allowances to start selling this at a large scale.
New Haven in Connecticut is the home of the prestigious Ivy League Yale University. Presidents have gone to school here, including George W. Bush and Bill Clinton. Looking for new phages is an integral part of Ben's research. At this sewage plant in New Haven, he found the phage that targets MRSA, a superbug that most commonly attacks the lungs and airways of cystic fibrosis patients.
I think biology was always in the cards for me. One of the parents in my first grade class brought in a microscope and got to see microbes for the first time, and it was like mind-blowing. It still is mind-blowing. The possibility that maybe something I do somewhere in the lab could eventually help somebody, that's the main driver for me, I think. Like, I don't know that we directly could manufacture enough phage to help millions of people. But I think the ideas and the concepts that we establish could be industrialized and really have a larger impact.
Yale is one of the leading institutions for phage therapy and research. The center has successfully treated about 100 patients through compassionate use and clinical trials.
We treat a lot of prosthetic joint infections. The individuals come to us as a last chance to try and save their limb from amputation. Had really solid success, nearly 100%. We're testing bacteriophage therapy in people that have cystic fibrosis. Compassionately, they work really well. 99.9% of bacteria killed.
After years of waiting, cystic fibrosis patient Maggie finally got approved for compassionate phage treatment. Superbug MRSA infects her lungs and airways, making it nearly impossible to breathe. Let's hope that Ben's superphage will do the job.
Okay. I've been getting to know you through your sputum, over time. And text, of course.
I love that.
So there's more meaningful stuff.
Thank you. Pardon my reach. May I? Thanks.
So this phage particularly was effective against your strain. We pulled it out of sewage, and we found a lot of cool resensitization to antibiotics when it became resistant to it.
Mm-hmm.
So hopefully that happens here, but obviously we have no idea, right?
Yeah.
Because you're at the edge of what we know.
Before we do phage therapy, through this compassionate use mechanism, we're always making sure that you feel that you're informed of the risks and benefits. This is really just an experimental therapy that we're adding into your clinical care because the FDA feels it's appropriate given the resistance to antibiotics.
Should she alternate her breathing in between her nose and her mouth?
We just have to make contact with the phage and the bacteria. So, you know.
The meat.
Yeah, the really deep breath, the better.
Deep breaths.
Yeah, deep and slow. And there you go.
Perfect. Yeah, great. Beautiful.
How fast does it start working?
14 days after you start. And that's when we anticipate seeing a large change.
So maybe you'll be sleeping in 14 days, you'll cough less, you'll feel less fatigue. I know, it's a wish and a dream.
We'll see, we'll see.
You're always so calm.
We doing this?
Yeah.
I mean, you guys are helping me.
I'm so attached to you, so thank you.
Yeah, anytime.
I rely on you so much, so.
Good.
Feel free to reach out.
I know. Appreciate you so much.
Yeah, of course.
Thank you.
After a few rounds of treatment, the phages break down the superbug, which can now be treated with antibiotics. With the infection finally gone, Maggie graduated from university with a degree in communications. Uganda is one of the most biodiverse countries in the world. Its capital, Kampala, is a hub for academic discourse and has one of Africa's oldest and most prestigious universities. Makerere University plays a pivotal role in educating and training the next generation of leaders and innovators.
It is not 15, my dear. This is 15, isn't it? Yes. These are 2 decimal points. You multiply by 100 by 100, you get 1,500. Yeah.
Professor Jessica Nakavuma is the leading researcher for phage therapy in Uganda. She founded Phage Team Uganda, Working with undergraduate students, she is searching for solutions to combat AMR for human health, animal health, and food safety.
For phages, we have our specific requirements. Don't do the experiment once. You repeat thrice. The undergraduate students, they want to finish their research. And we find ourselves we cannot publish that work until we redo it ourselves, which is a waste of resources. And you know we are resource poor, so don't just do research in order to get this immediate award. Let us not be short-sighted. We look at a larger picture. Growing up, we could hear of death in your village, maybe one person and a very old guy or lady dying. But it is very common these days, to the extent that the young kids no longer fear dead bodies. We need to have a solution as soon as possible, maybe as soon as yesterday. As I was told, there are bureaucracies involved.
Professor Rita Nakayinga is also part of Phage Team Uganda. She visits the sewage plant in Kampala with her colleagues for another phage hunt.
I thought you were taking the other side, Abraham.
The other side? Here. It was here, I remember, because we... and...
Abraham, don't fall.
No, I won't. They are in the middle. In the middle, that is where you will find We started in 2020, a project that was funded by UNESCO for us to hunt for phages that could kill bacteria that infects the banana, and the banana being a staple food crop here in our country. Have you tried the banana though? The matoke. But most people say it's flat. It has a flat taste, it's plain. But for us, it has also cultural connotations to it. Current methods that are being used to manage the disease are still ineffective. So they still use antibiotics, they use copper-based biopesticides, they use agricultural practices by cutting the plant or burning the plant or burying the plant. But they seem ineffective because we keep getting all these outbreaks, continuous outbreaks of the disease. So now we thought, okay, maybe it's best to try an alternative, which is the phage technology, what we bring over here to our incubators. So this is the phage in what we call an enrichment solution. The sewage is enriched so that the bacteria can grow, and then the phage is able to infect them and produce as many phages as possible. Here is a nice little phage, the head, and that is the tail. The first one has come from the sewage plant. It's a superhero to us because it's the first phage that our lab has identified, and it excites us. I mean, it's the first time we get to see how a phage looks like. It's the first time we get to see what a genome of a phage looks like, but it only kills 4 strains out of 26. So that means that because of that, we have to keep on looking for that superhero who can kill as many 26 and beyond strains. And if we can get that, boom, we will have hit the goal. Yes.
There are plans to build a phage bank at Makariri. Most phages need to be stored at -80 degrees. Constant power outages seem to make that impossible. But the scientists here don't give up that easily. There is always a way.
So come in. As you can see, all these facilities have restricted entrance and restricted exit. So it actually caters for both safety and security. There is a lot of quality assurance that is done in a biorepository. As you can see, even the nature of the cables, the power is tapped from above. Because these things defrost a lot, and they release a lot of water, so you can easily create an accident in the biorepository. There is a system that can alarm, it SMSs you, it texts you wherever you are, that the temperatures are fluctuating or there is a power problem. Some people think biorepositories are very dangerous, but they are not dangerous if they are being handled by professionals.
Foodborne infections are a significant health problem. A recent report indicates that 11% of foodborne pathogens are resistant to antibiotics, and 36% are multi-drug resistant. In neighboring Kenya, phage therapy is already in clinical trials at the International Livestock Research Institute in Nairobi. This is urgent research, as 60% of all mammals worldwide are livestock, raised to feed mostly one species, humans. The overuse of antibiotics in livestock has reached alarming levels. In an international partnership with Laval University in Canada, Angela Makumi and her team are conducting a clinical trial using phage cocktails to treat salmonella-infected chicken.
In short, I was a molecular biologist, microbiologist. I studied a lot on phages over 12 years. So first of all, we infected them with salmonella. We watched how the salmonella is reducing over time after different intervals of phage treatment. Now we put it in water, and then we let them drink. Inoculate again. Water. 400. It's not salmonella for chicken, it's salmonella zoonotic for humans. And so it won't make the chickens very sick. Just to reduce the amount of salmonella that gets into the environment or gets into the slaughterhouse by using phages. We can bring another chick. Hush. Sorry. Contaminated. I'm contaminated.
Sorry.
I'll not be there when it flies.
He's afraid of chicken.
It's not only a problem for developing countries. Very soon it's going to be a problem for livestock. It's going to be in the environment. It's going to also be in the humans.
In collaboration with this biotech firm, salmonella phages are added to the phages from Ilri in Kenya for the chicken cocktail.
We dehydrate the bacteriophage to make them into a very concentrated powder. You can add 1 gram of this powder in 1 ton of feed, and that will still be efficient.
This is the powder, so we've spread it. So we analyzed it. We added some protective agents in there, so some sugars, some proteins. They're still active in this powder after the spread drying process.
You could make lemonade.
Yes, good. Lemonade with Phages.
Prevention is key in that livestock industry. 85% of the antibiotics produced worldwide are used in the livestock industry. Only 15% are used to treat humans. When there's a strain that is problematic in the livestock industry, it's 95% resistant to antibiotics. So we need to move away from using antibiotics as a preventive treatment.
And we don't have to, we have to start now.
Yeah, we should have started 10 years ago.
What looks like an experiment in its infancy at this startup in Quebec City has reached another dimension in Columbia, Maryland, at the biotech firm Intralytics. Intralytics specializes in the development and production of bacteriophage products for food safety and human health. Several of their commercialized products target foodborne pathogens such as Listeria, Salmonella, and E. coli.
We have a 1,500-liter fermenter. We typically use it at about 1,000-liter operating volume. We end up with maybe 50 to 100-liter of highly concentrated, very pure, pharmaceutically or food-grade acceptable buffer that will go into the storage until we are ready to mix and formulate the final cocktail. It is a fermentation process, yeah. Like a beer, you're making a beer, right? I was born and grew up in a country called Georgia where phages are commonly used. Almost everybody I knew have taken them. You can imagine the pushbacks we experienced when we formed the company. Nobody knew if we could patent phages or this technology to have some commercial success. We were the first company in the world to receive FDA approval for food safety product. Made news all over the world because FDA allows viruses to be put on food. It's 100% natural, it's environmentally friendly. Most of our products are also certified kosher, halal, and organic. I feel like we are having some significant impact on human lives.
Despite FDA approval, many companies, like this dog food producer, prefer to remain anonymous. They fear repercussions if they openly admit to using viruses in their products.
This particular bottle here has Samafresh. That's where they spray onto the meat. Millions and millions of little phages.
Lake Victoria is the largest lake in Africa and a vital source of livelihood for millions of people in the region. However, the lake's fish stocks have declined due to overfishing, pollution, invasive species, and climate change. To protect the declining stock, fish licenses are expensive. Those who can't afford them fish illegally, but most days, the locals are not successful. To satisfy the increasing demand of a growing population, aquaculture is seen as a viable solution. Now, more fish are grown in fish cages. But the environmental impact is immense. High-density farming causes an increased risk of disease transmission among farmed fish. The use of antibiotics in the feed is high. and many pathogens are now multi-drug resistant. Fish farmer Thomas Misisoke is part of ongoing research into phage therapy for salmonella infections.
These are my nurseries, Nancy.
All the runoffs.
All around. And this water comes from about 2 kilometers away from here. So I'm in the middle of the valley. Yet I've got a license for fish farming.
Kakati, what we are going to do is we shall need the pond sediment after collecting the fish.
How are you going to get the fish?
You don't have a net, a seine net?
He doesn't have.
Some farmers use chicken waste and this and that and that. But in chicken waste, there are some residues of medicine and everything, so which is harmful to what? To people. But me, I want to make it as safe as possible. I don't want to poison people, I want to keep people alive. Why I'm bothering scientists? Because I read, I go for seminars, but I'm not a scientist.
Is there fish here?
There are very many.
Because these ones are reproducing. Okay.
Have you seen any fish dead recently? Any fish floating dead?
Something? No, I can't allow that.
But if they die, they die.
If you see anything that is misbehaving, I would like to take a look on it.
Okay.
Only 9 kilometers from the city center, the Nairobi National Park is home to many wild animals. Kenya is one of the most popular safari destinations in the world, attracting millions of visitors each year. Safari tourism supports the conservation of wildlife and their dwindling habitat. This is an essential investment since humans destroyed 83% of wild mammals. However, safari tourism also has its downsides. Windy, impenetrable forest in Uganda is one of Africa's oldest ecosystems. It is home to half of the world's endangered mountain gorillas. Wildlife conservation is taken very seriously here. There are many local, national, and international collaborations to protect the endangered primates. Nelson Bakamba is a wildlife veterinarian in Burundi. Concerned with the potential threat of antimicrobial-resistant infections in wild gorillas, he started his preliminary research into phage therapy.
Salmonella is a very hardy organism, very resilient in the environment. It has been reported to cause severe diarrhea in both humans and gorillas. And there have been cases of gorillas that have died from salmonellosis from zoos. Gorillas are not going to be looking for antibiotics from any shop around, but how they get to have this? In Burundi, we have over 20 mountain gorilla groups surrounded by rural, peri-urban areas, mini towns that are cropping up because of tourism. We have rivers crossing through the park, coming all the way from communities of which gorillas drink from this water. We have baboons around Burundi trying to run away with foodstuffs from people's homes, and they go back into the forest. Every day, someone travels across the globe to come down to Burundi. That increases the chances of introducing antimicrobial resistant strains into the area. The gorillas, we have now about 1,000 individuals in the whole world. Any single threat, any single risk of disease or of infection can be devastating to such a population. The wildlife populations, the human populations, and the livestock, and the environment itself as a whole. Everything is connected. Wild populations, most of its survival is going to be determined by our lifestyles as humans.
Mountain gorillas are closely related to humans, sharing 98.4% of the same DNA. This makes them highly susceptible to human diseases. Once, humans killed them with machetes. Soon, we only have to get close to the gorillas, and our relatives might die of a simple infection. We are back in Kenya. The Institute for Primate Research is hosting a symposium on phage research. Some stakeholders in public health will also be there. Can they be convinced to support this new medicine?
We're hoping that KEMRI will become have a national phage bank, a collection of phages that could be quickly deployed in an outbreak. What I'm going to talk about today is sharing information from regulatory agencies so that they can start thinking about how do you want to regulate phages.
Thank you for having risen up to give us a very interesting talk. Africa and the world is rising up to AMR. The only hope is now the phages, meaning this is where we're going.
Yeah.
Last time we had the phage meeting was around
2018 in
July.
June or July? Yeah. Yeah.
Talking about the future of bacteriophages.
Yeah, exactly.
Yeah.
Exactly.
It took time, but
from this small lab you see here, we have come up with many publications. And from the same lab, we are hoping to come with a commercial phage product. This is our research team. Most of the students were trained during the phage training program that Toby was leading. So Toby, we are always grateful. We hope you are planning to bring another one.
I am.
Because we really want to expand this program. Very soon you will see patients being treated.
Yeah, I actually think there's a possibility the first patient might be treated by the end of the year based on our meetings with the regulators yesterday.
Yeah, and we hope to be the first in Kenya, in Kenya and in Africa, at large. So it will be an example. And Ben here promised to come and help. We shall move it from here to the patients. That is all we want to do.
And how many phages do you think your team has isolated in total so far?
Approximately 120 phages. So we are looking forward to compassionate use because the phages that we do have, they are very potent. Most potent in host range, and then again in size, when we look at the lysis or the plaque size, a very vibrant phage that maybe one plaque can actually lyse the whole plate.
Interesting.
Yes.
And then the great news. Ben and Giuliani have won approval to add phages to the water supply in Kilifi, Kenya, to prevent bacterial outbreaks.
It's so exciting for me that it's a project developed in Kenya. You guys are leading the way.
For sure.
Yeah, you're not depending on some stupid company from New York or from England or something. You guys are doing it. You're making it. Right? In Kenya. It's the first one I heard from yesterday.
In essence, and there's so much that we can do, including the vaccine development from the phages. So that is the thing.
Yeah.
We can't wait for it to happen.
Yeah.
And when we deliver that, nothing stops us from doing anything.
Yeah.
Our phages are able to lyse bacteria from outside Kenya with that efficiency of about 100%.
Yeah.
So we are going to export it instead of importing it.
Yeah.
I think as we go on, we will simply just wake up from our beds, go sample, see the bacteria, and fish the phages from the library, and we're able to do it.